Good day everyone, today we will present chapter 6 cell division from the Form 4 biology syllabus. To understand cell division better, we must understand structure called chromosomes. Chromosomes are thread-like structures located in the nucleus and are made up of DNA and proteins. One set chromosomes originate from the male parent and another from female parents. DNA carries genetic information that determines the characteristic of an organism. So the first stage is interphase, which is the preparation stage. The second stage is M phase, which includes mitosis and cytokinesis. The purpose of the cell cycle is to ensure that genetic information is accurately distributed to the new daughter cells.
Cell division is the process in which a parent cell divides to form new daughter cells. Before a cell divides, it goes through a stage called interphase. Interphase is the longest phase in the cell cycle, where the cell prepares itself for division. During interphase, three main processes occur. First, during G1 phase, the cell components grow in size such as mitochondrion and endoplasmic reticulum. At this stage, the nucleus looks big and the chromosome is in the form of chromatin. Second, during S phase, the DNA replicates, producing identical copies of chromosomes known as sister chromatids. Both chromatids are joined at the centromeres. Third, the G2 phase. The cell will continue to grow active, gather energy, and make final arrangements to enter the next stage of cell division. After all the preparations are completed, the cell proceeds to cell division through either mitosis or meiosis, depending on the type of cell and its functions.
Mitosis is a type of cell of division that produces two daughter cells that are genetically identical to the parent cell. Each daughter cell contains the same number of chromosomes as the parent cells. Mitosis occurs in somatic cells, which are normal body cells such as skin cells, muscle cells, and nerve cells. Mitosis occurs in several stages. First, where chromosomes condense and become visible while the nucleus membrane begins to break down. Second is metaphase, where chromosomes line up along the equatorial plane of the cell. Third is anaphase, where the sister chromatids separate and are pulled to opposite poles of the cell by spindle fiber. Fourth is telophase, which nuclear membrane reform around each side of chromosome. Finally, cytokinesis occurs dividing the cytoplasm and producing two identical daughter cells. Mitosis is important for growth, repair of tissues, and replacement of damaged or dead cells.
Meiosis is a type of a cell division that occurs in reproductive organs to produce gametes, which is sperm and also egg cells. Meiosis occurs in the testes for the female and also the ovary for the female. And this meiosis process, which will be divided in two processes, which is meiosis 1 and also meiosis 2. Let's see how does the process occur. During prophase 1, chromatin shortens, thickens, and forms visible chromosomes. The pairing of homologous chromosomes, which is synapses, will form swivelant or known as tetrad. Crossing over process will also occur. And also, crossing over produces a combination of genes that are new in chromosomes. At the end of prophase 1, the nucleus membrane and nuclei will start to disappear. Both centrioles will move toward the opposite pole cells, and also spindle fibers are formed among the centrioles. After that, during metaphase 1, homologous chromosomes are arranged at the equatorial plane. Also, sister chromatids are still tied together because the centromere has not separated. During anaphase 1, spindle fibers will contract and cause each homologous chromosome to separate from its homologous parent and be pulled to the opposite poles. Last but not least, during telophase 1, the chromosome will arrive at the opposite pole cells that produce two daughter cells which are haploid. Alright, for meiosis 2, we will start with prophase 2. In prophase 2, the nuclei and the nuclear membrane will disappear, and also each chromosome will join at the centromere. The spindle fibers will also start to form in both daughter cells. Next, in metaphase 2, chromosomes are arranged at random on the equatorial plane for each daughter cell. Each chromatid is tied to the spindle fibers at the centromere. After that, during anaphase 2, the sister chromatid centromere will start to separate. Sister chromatid will separate and move towards the opposite poles. In telophase 2, chromosomes arrive at the pole of the cell. Spindle fibers start to disappear, and nuclear membrane and the nucleoli are reconstructed.
Both mitosis and meiosis are important processes, but they have different characteristics. In mitosis, one cell division occurs and produces two identical daughter cells with the same chromosome numbers as the parent cell. In meiosis, two cell division occurs and produces four daughter cells, each with half the chromosome numbers. Mitosis occurs in a somatic cell, while meiosis occurs in productive cell to produce gametes. The importance of cell division includes: first, it allows organisms to grow and develop. Second, it helps repair damaged tissues and replace old cells. Third, meiosis ensures sexual reproduction and genetic variation. In conclusion, cell division ensures the continuity of life by producing new cells and passing genetic information from one generation to the next. Thank you for listening to our presentation. Goodbye, everyone.